Hello and welcome to today's session, everyone. Today, we are truly honored to welcome Dr. Katalin Kariko. Katalin Kariko is a professor at the University of Szeged, an adjunct professor of neurosurgery at the Perelman School of Medicine, University of Pennsylvania, where she worked for 24 years. She is the former senior vice president at Biotech, Germany, where she worked between 2013 through 2022. She received her PhD in biochemistry from the University of Szeged, Hungary in 1982. For 4 decades, her research has been focused on RNA-mediated mechanisms with the ultimate goal of developing in vitro transcribed mRNA for protein therapy. She investigated RNA-mediated immune activation of RNA and co-discovered the nucleoside modifications that suppress the immunogenicity of RNA. With this, she widened the therapeutic potentials of modifications of mRNA. She is a co-inventor of mRNA-related patents for the application of non-immunogenic nucleoside modified RNA. 14 of those are granted by the US. She co-founded, and from 2006 through 2013, she served as CEO of RNA-RX, a company dedicated to developing nucleoside modified mRNA for therapy. Her patents, co-invented with Drew Weissman on nucleoside modified uridines in mRNA, is used to create the FDA-approved COVID-19 vaccine by Biotech, Pfizer, and Moderna to fight the pandemic. In the last 2 years for her achievement, she received more than 100 prestigious awards, including the Japan Prize, the Horwitz Prize, the Paul Ehrlich Prize, the Benjamin Franklin Medal, the Canada Gairdner International Award, the Kovalenko Medal, the Tang Prize, and the Warren Alpert Prize, the Princess Asturias Award, the BBVA Frontiers Award, the German Future Prize, the Breakthrough Prize, and the Lasky DeBakey Clinical Medical Research Award. Of course, I'd like to thank our audience for joining us for the special event of welcoming distinguished Dr. Katalin Kariko, co-discoverer of mRNA-related patents to develop the highly effective COVID-19 mRNA vaccines by BioNTech, Pfizer, and Moderna. We'd like to extend our deepest appreciation for your innovation and time today. Now to Dr. Katalin Kariko.
Thank you very much. Sorry, Gisela, that so many special words were there in the CV, but I am very happy to be here and talk to the audience here about a little bit about how I, learn and how develop the interest to work with the RNA, mRNA, and how all of this evolved and where is this field right now. And so for this one, I prepared a presentation, and I will share my screen. Okay. So can you see?
Yes.
Okay. So I will talk about how this mRNA was developed for therapy and vaccine. And as the subtitle says that I am an immigrant, I say here that okay. So I am from Hungary, and I grew up there behind the Iron Curtain in the 50s and 60s. My parents were not formally educated, very smart people, but had no chance to study. And my father was butcher, my mother was a bookkeeper, and I had a sister who was 3 years older. And as we went to school, of course, she was very good, and then I had to be good also. And eventually, it's a small town, 10,000 people, and both of us dreamed about going university, and eventually my sister actually got a PhD in economy. But my interest was in biology, and I was dreaming about to go to University of Szeged, because right next to it was this biological research center, and I successfully was accepted there. It was very difficult to be a biologist. There were like 20 was accepted from the whole country. And when I was undergraduate, I started to go to this biological research center. I want to do some kind of research, but the only opening was actually in the lipid team, and the supervisor said that, oh, rest of your life you can have in the bench during the summer, go and do some experiment in the Fishery Institute, where you have to collect fat from fish, and we will determine which kind of chow material they are eating and so on, and whether, you know, if you have a fish on fed with corn, will be like, you know, the fat will be like pig fat. Anyway, I did this first experiment here, and when in the fall I was still undergraduate student, returned to the lipid lab, you know, where scientists were approached us that they needed phospholipids because they want to make a liposome. And we are in the 70s still, you know, I was still student, undergraduate student. And so we isolated phospholipid from the cow brain and did this first experiment where we delivered plasmid DNA with liposome wrapped into the mammalian cells. And this is just important because I don't really comprehend it fully, that how revolutionary we are doing and what we are doing, but important that, you know, to start with something and make your hands wet and think about something. So in the lipid lab, you know, one day just a colleague walked in who was the head of the RNA lab, and then he said, oh, okay, you can come to do PhD in my lab. So this was no rotation, not a formal way to do, but it was Jenny Thomas, and he was organic chemist, and he was heading the RNA lab. And actually he did synthesize different kind of cap analog in the 70s and send it to Aaron Shotkin here in New Jersey to identify what is the cap structure of RNA. But my responsibility was to synthesize this small molecule, which was we are in 1978, so the prior year was discovered, this 2'5' link oligoadenylate. And here I worked with János Ludwig. He was a fellow graduate student, and I am working with him even today. So that, again, a very important thing that, you know, to be good with your fellow students, because you will need their advice later on. So we synthesize chemically and enzymatically this small RNA molecule. And this is interesting because it is 2'5'. And so for my studies, actually, I used the 3-deoxy analog, which called cordycepin. So my thesis was about this nucleoside analog 2'5A molecule. This molecule is actually naturally produced in your body when a double-stranded RNA is present. Oligoadenylate synthase is producing it, and then this small molecule can activate RNase L, this latent RNAs, which can chop up a single-stranded RNA. And this was in the 70s with when it was discovered, considered that it is an interferon related antiviral mechanism. And if you followed the SARS COVID-19 research, you could see that those people who had low level of oligoadenylate synthase enzyme, as actually they get seriously sick from when they get infected. So even it is also important, I am not working on this field anymore, but still follow and get excited when I learn new things about these old molecules, of course. And I came to the United States and worked at Temple University, and we still worked on 2'5' oligoadenylates, but when in this time in 1985-86, the major problem was HIV, and we couldn't make this antiviral molecule feasible for therapeutic use. So we use a double-stranded RNA and try to induce interferon in the patients. I have to say interferon was discovered in 1957. Lindemann Isaac discovered, and by 1962, we knew that double-stranded RNA can induce this interferon, which interferes with the viral replication. Interestingly, when we used here in 1986, 87 in a clinical trial, we still didn't know the mechanism up until 2001, when we learned that double-stranded RNA activate toll-like receptor 3, and that's the mechanism in using interferon. Anyway, the double-stranded RNA was very toxic, so we used this mismatch form and performed clinical trial together with the Hahnemann University. Unfortunately, the patient could not make interferon and was not beneficial for them, this trial, because they had anti-RNase L inhibitor which was induced by the virus. So I was talking about short RNA, double-stranded RNA, and so on, but did not mention messenger RNA. By that time, in the 80s, it was very well known, that messenger RNA was discovered in 1961, and two papers published this in Nature, and both of them mentioned that this is a very unstable intermediate. And this was from the 50s already was looking for this magic molecule, because they did not understand that how the information is getting from the chromosome, which is present in the nucleus, to the cytoplasm, where the protein synthesis factory and the ribosomes exist. And with that discovery, we learned then the information flow is DNA, messenger RNA, and protein. So of course, the 61 was a very exciting time, because it was published in May. In the same year was already solved the code, like at least two of them. The phenylalanine was UUU code, and the CCC was for proline, and there many other things happened in 1961. But as with all of the discovery, I can say that following the discovery, there is a phase which in mRNA lasted for 20 years or more, when structurally and functionally analyzed the molecules. And it was, you could say, the same thing for the CRISPR-Cas9 system. So that is characterization. Then scientists start to, or forming small company maybe, and they start to see that whether it is their discovery can work in animals, and when they have some success, then they start to use in human trial, and eventually they get an approved product, which I will talk about. We don't have here one thing, which is, acceptance by society, which is also a very important part as we learn for the COVID. So let's start what happened, when messenger RNA was discovered, and then started to analyze this structurally, the RNA. And early on, they discovered 1970 already that the RNA has a poly-A tail. And soon they introduced a method, oligo-DT purification. So then messenger RNA could be purified and used for different kind of analysis. In the 75, this purified RNA was already used to see that how the other end of the RNA looks, so that they discovered there is a very interesting cap structure is there. And in 1975, several Nature papers characterized and demonstrated importance of this cap structure for translation and other function. By the end of 75, enzymes were discovered that viral enzyme can do and can enzymatically, they can cap messenger RNA. So this was the structural characterization. Other scientists started to isolate RNA, and this they reached out for this reticulocyte, which is the precursor of red blood cells, which is loaded with beta globin, and they could separate on columns at the beginning by weight, and then because beta globin is so, and mRNA so enriched in these reticulocytes, so they could see on the chromatogram, isolate, and then translated in rabbit reticulocyte. They injected in frog oocyte. This was a huge cells, and in microcapillary can be injected. If you would be at my age, you would know that if you work in a genetic lab, you have seen a terrarium. There were a few frogs who were there. And more importantly was that Oleg Bankham discovered and described in London how to make liposomes in the 60s, and some scientists took this globin, beta globin mRNA, and put in liposome and delivered in mammalian cells, and they could demonstrate that the protein was made. So this was the first time that mammalian cell, the delivered messenger RNA, could be delivered. The problem with this one was that only isolated RNA could be analyzed. And so the beta globin mRNA was the only one. So a big step was needed, and this was delivered by Harvard scientists who introduced, again, I am saying these scientists, even before them, scientists try to use bacterial RNA polymerases, whereas here is described the phage RNA polymerase, SP6 RNA polymerase, and 1985, the T7 RNA polymerase. These are different phage RNA polymerases. The phage is the virus of the bacteria, and they have transcribing RNA with high fidelity, very quickly, very processive, and a promoter is very short sequence, and so it was very feasible. And the first what they transcribed from DNA sequence was messenger RNA coding for human beta interferon, and they kept it actually with Bernard Moss gave them the capping enzyme from the Suboxenia, and then they injected this to frog oocyte. And when this mRNA was injected and cultured overnight, then the medium contained human interferon, which was functional. So this was open up a big opportunity for making more and more different kind of mRNA. And it is 1980, the second part of 1980s comes, the PCR, the TA cloning, the simplified RNA isolation. So it was a revolution, and so many people started to work on messenger RNA in the 90s. And they delivered vasopressin mRNA, for example, the script, and they treated model animals successfully. They started to use in vaccination for infectious disease, for influenza, and also as a cancer vaccine. But if you look at in these scientists and these papers, you will find that they were not publishing more paper than these, because they run into a problem. And the problem was that the RNA was very fragile. It was translated very short period of time, and very small amount of protein was produced. And later on, we also showed that this RNA was immunogenic. So it was somehow it was not very appealing. And in addition, in the 1990s started the Human Genome Project, and when they identified a genome and responsible for some diseases, gene therapy was in the forefront, and those were all with DNA based. So that was the problem. And but there were some people like me also started actually in the 1990s making RNA and in vitro transcribed RNA, working with the cardiology department, and very kindly those scientists at Harvard send, in an envelope, send the plasmid, and then I could make RNA and clone into different things. We were interested in urokinase receptor. Why I'm showing this example, which we have done, because when we delivered the mRNA to a cell, it was interestingly that this protein for function required so much post-translational modification, carboxy terminal, and processing. It somehow the cell knew what to do and then created this functional protein. And I had to move, and several times I had to move in life. I didn't mention, but terminated in my position several times, and otherwise I wouldn't be here. So I moved from cardiology, that I had to move somewhere, and then it was actually David Langer, a colleague who I met in a cardiology laboratory, and he was a medical student like you guys are, and he was a bit obnoxious, but then he was very much interested in what I am doing. And when I needed a new place to work, here is my laboratory for 17 years in neurosurgery. David was neurosurgery resident and convinced the chairman to give me salary and give me a lab space, and I did all of this nucleoside modified RNA and everything, and work with David ever since. So this was, we tried two different messenger RNA we generated, and coding for different proteins, try to come up with a solution for stroke therapy. One day I ran into at the Xerox machine really to Drew Weissman, and then he was interested to develop vaccines for infectious disease. And so I made the RNA, what he needed. And when he used human dendritic cells, these are the antigen presenting cell, professional antigens presenting cell. And we are in the 1998 when we met. And so he used this RNA, and then he demonstrated that the RNA I made and delivered with lipofectin at that point was very inflammatory. And up until that, I was not worried about any immunological reaction. But here we are. I want to use this messenger RNA in treating a stroke patient, and it is very inflammatory. So with Drew, we started to think about why it is inflammatory. And because we have mRNA everywhere in the cell, but of course, to these immune cells, the RNA is coming from outside, and maybe out of the cell, the RNA may be inflammatory. And so we decided that we investigate what kind of mechanism is responsible for that. So we isolated RNA to test out that whether really in our cells, mRNA and different RNA in our cells is also inflammatory. We isolated different kind of RNA from mitochondria, nuclear, and messenger RNA, as well as bacterial RNA, and exposed these dendritic cells, and we measured TNF-alpha. And here in purple, you see that TNF-alpha induced by the in vitro transcribed RNA, and the isolated RNA was not as inflammatory. What was interesting is that the tRNA was not causing any inflammation at all. And knowing that the tRNA has so many different kinds of nucleoside modification, actually 25% of the nucleotides there has some modification, we were thinking that could it be that these modifications somehow suppress the immunogenicity and inflammatory nature of the RNA. So we look at there, and there are more than 100 different modifications exist, and we were wondering that which was important and how we could prove it, because how these different modification incorporates in our RNA. All of our RNA is made from the 4 basic nucleotides, and post-transcriptionally modification introduced, but most of the enzyme was not known, so we couldn't just order and test out. So I mentioned here that one of the, or two of the modification, the 2' methylation, so methylated on the ribose here, and also the pseudouridylation, how it occurring naturally in our ribosomal RNA was discovered by a fellow student from Hungary, Tamás. We lived in the same dormitory. We worked at the same institute later when we finished our studies, and he knew. So I called him and asked for help, although he could not provide all of the components, and anyway, certain sequence was required for that. But it is important message for you, just to follow your colleagues, your fellow students, which field they are going, because one day you might need their help. And if you don't follow, you don't know that they are expert on a specific field. So anyway, we couldn't make RNA on the way as nature is doing. So we decided that we will purchase modified nucleoside triphosphate and try to see that whether the T7 RNA polymerase will incorporate and we can have RNA. So when we selected only naturally occurring nucleoside triphosphate, and so then we found that out of the 10, 5 could incorporate. Of course, we didn't know at that point, is any of these important for suppressing immunogenicity, but this is when you do basic science, you don't really know what you are doing, you just expect that it will be successful. So when we made this we isolated these synthetic RNA and exposed to dendritic cells, we found that some of them were not inducing any inflammatory molecule. And when we looked at closer, we found that, oh, those which not inducing, those every case in the uridine was somehow modified, was pseudouridine, 5-methyluridine, or 2-thiouridine. So it was we were very happy that we found the non-inflammatory RNA. So incorporating these to the RNA, not inflammatory, but of course we want to make mRNA coding for a protein. So it was also critical to test out whether, if we translate, whether protein is produced. And from 5-methyluridine, we found the same amount as from the conventional uridine. The 2-thiouridine did not produce any protein. So the mRNA containing 2-thiouridine, no protein, but the pseudouridine, we get 10 times more protein, which was very surprising. And I spent 3 more years just to try to understand what is the reason for that. And as it happens, after all of this discovery, or even prior to that, we filed a patent and we established a company, RNARX, and submitted a small company, to get some money. And we proposed that we will generate nucleoside modified mRNA encoding erythropoietin, and we will treat animals with anemia. And so this is what we have done subsequently. And we demonstrated that the RNA was modified, pseudouridine containing RNA, very small amount, 0.1 microgram was sufficient, that when we injected a small amount of protein, we still could detect 4 days later. The EPO, erythropoietin half-life is about 2 hours. So if a big bolus you inject, 6 hours, you still can detect the protein, when you inject the protein, and then disappears. And when you containing RNA, it also disappeared much faster. And when we injected, we could see the biological effect. We could see hematocrit increased by day 6 and 7. And then when weekly injected a small dose of EPO mRNA, this is murine-specific EPO-RNA, we could see that the hematocrit was maintained. And most importantly, this RNA did not induce any inflammatory molecules. So with that, again, I had to pack and then moved again and went to Germany. At that time, we didn't have this nice building. We were on the campus, and here, together with Biotech founders, we try to further improve optimize the RNA. We did codon optimization, the different untranslated region optimized. We did purification and introduced more nucleoside modified RNA. And then eventually what we could achieve, that what I mentioned in the 90s, this small amount of protein produced from the injected RNA short time. Now we had significant amount of protein, significant amount of time. So we were ready for exploring further it. So there I participated in the first project was that we generated messenger RNA coding for bispecific antibodies. And these are recognizing CD3 on the T cells, and this was Claudin 6 is a tumor-specific protein, and it brings the bispecific antibody close proximity to components, and we demonstrated in animal models that the large tumor could be eliminated. This was in 2017. This is already in a human clinical trial, this same molecule. We also generated a conventional, not bispecific, but conventional antibody. These were Claudin 18.2 specific. Those are also in human trial. Another project, we did intratumoral injection of mRNA, where cell surface close tumor here in a melanoma model is shown for animals. These messenger RNA code for 4 different cytokines, IL-12, interferon alpha, GM-CSF, and IL-15 sushi. And the principle here is that locally this tumor is injected, and then these molecules will invite immune cells to close to the tumor, and those immune cells, when they are circulating, they recognize the remotely located tumors and this upscopal, so-called upscopal effect, and eliminate metastatic tumors. And here we showed how it was eliminated from the lung. And this is also with the Sanofi is going in undergoing clinical trial for 2 years now. So my goal was always, as I mentioned, to develop mRNA for therapy. So here at Biotech, I was responsible for the protein replacement therapy. And vaccine is my colleagues' main interest was. So what happened is, and we are all familiar with this, that 1961, I told you that RNA discovered, and 60 years later, we had the first approved, FDA-approved mRNA in the form of this vaccine. And so what happened in that 1 year, in 2020, we already know that in January, we learned the sequence information from colleagues, and mRNA companies started to make a vaccine, and not just RNA companies, other companies, like 150 different vaccine developments started. I have to say that if it happens 20 years earlier, sequence information would not be sufficient. Those who want to develop something, they needed a little package which contains the biological material for it, because it was 1999 when first company was established to synthesize genome. So so many different fields had to be developed so that we could see this kind of progressive development of the RNA vaccine. And so if you follow during 2020, just in the TV news, you already were well informed what happened and how we, by December on the same year, we could have the vaccine. So many people thought that this was the first time that nucleoside modified RNA was used, but if you were in our field, you would visit our mRNA therapy meetings, you would know that well before that messenger RNA was used. I could say that messenger RNA was used as a vaccine in 2015 for CureVac made mRNA for therapeutic as a prophylactic vaccine for rabies. There were Moderna had Zika viral vaccine. Moderna had a vaccine for influenza. And those were human trials. It was just, small scale and not what we have seen in the case of SARS COVID-19. And for example, therapeutic use of mRNA, nucleoside modified RNA for VEGF-A started in 2018. So it was well before the pandemic. And this is still an ongoing, the phase 2 is finished, but still this trial is ongoing. This is VEGF-A is a mRNA which generate new protein, the coded protein generate new blood vessels, and this was used for patients undergoing bypass surgeries. And then these as a naked form were injected to the heart and increased the vessel in the muscle of the heart and improved the patient life. And another thing was also successfully use nucleoside modified RNA for patients who are struggling with amyloidosis. These, after diagnosis, these patients usually die in 5 years. The liver is making a toxic misfolded protein, and using the CRISPR-Cas9 system, these patients, they deliver mRNA coding for this Cas9, the editing enzyme, and delivering the guide RNA, and they could successfully interrupt this toxic protein encoding gene in the hepatocyte of this patient. And this clinical trial is also ongoing, and successfully eliminated toxic protein production in this patient. And with this, we are here now in 2023, and if it's about like 150 clinical trials ongoing using messenger RNA. And the most of the trial is for infectious disease as a prophylactic vaccine, RSV, flu, CMV, those are in phase 3 phase. Flu is both Pfizer, BioNTech, and Moderna. Moderna has RSV for adults, CMV, yes. HIV, also a clinical trial. Zika virus vaccine, also for Moderna is using. Herpes simplex is BioNTech together with University of Pennsylvania, EBV, Nipah, TB, malaria. These are, so not just against viruses, but also for bacteria and parasites. For cancer, we know that messenger RNA coding for shared tumor antigen, there are FixVax program for the BioNTech, there are also antibodies. I mentioned the bispecific antibody and conventional antibodies against cancer-specific proteins. There are CAR T cells, there are intratumoral injection of cytokines, as I also mentioned already. For acute diseases, there are VEGF-A, and in addition to that, relaxin, also for heart failure. And VEGF-A was also used for wound healing, the VEGF-A mRNA. Of course, all of this is for mRNA in the trial. And VEGF-A for diabetic patient necrotic wound healing was accelerated, and many other diseases. These are all in human trial for urea cycle diseases, ornithine transcarbamylase, or other diseases, cystic fibrosis also start clinical trial. I cannot even say that how many different things is actually in a preclinical trial where animal studies were reported for tolerization, for in vivo CAR T cell generation, for heart fibrosis, allergy treatment for peanut allergies, and also interestingly for many things infectious disease. Some of them is like for tick related diseases is interestingly it's not against the bacteria or the virus which is carried by the tick, but they are generating in preclinical trial messenger RNA coding for 19 different tick specific saliva protein, and that is simplifying. So when the tick is biting the animals, actually they immediately reacting and preventing spread of the bacteria or the virus in the system. So there are many different applications are there, and with that, I would like to finish here. Yep. Stop sharing.
Great. We have a couple of questions that have come through.
Okay.
Thank you so much for that incredible presentation, Dr. Kariko. That was very interesting. One of our questions is, I'm curious, when you say that we take RNA to experiment, how do you manually replicate the RNA?
So the messenger RNA, what we presented here, is transcribed from a plasmid. It's about 1 microgram plasmid. You can generate 100 microgram of RNA. So it is this enzyme, this T7 phage RNA polymerase, is repeatedly just transcribing, and considering, that it's not just even 100 times, because, the RNA which produces single-stranded RNA, so it is about like 300 times is transcribed in one template. One plasmid template. So then we can have the RNA, purify, we remove the DNA so that only the RNA is present in the sample.
We have another question asking, your outstanding career path hit several bumps along the way. What did you do to remain positive and not quit?
So when I was in high school, we wrote letters to Hungarian scientists who are famous, and we couldn't speak English. In our high school, we couldn't learn that. And one of the scientists we sent letter, it was Hans Scheyer. And then he responded, and we were so enthusiastic. And actually, Hans Scheyer, he introduced the word in the 1930s, the word stress. And he described that the stress can kill you, but you need stress, positive one. And then I was 16 years old, and I learned about how to handle the stress. And that's what I can tell here, actually, maybe could be useful, because stress is everywhere. And how do you mean that we need stress? So that this morning I got up, I get excited, the anticipation we will be here. And so you need something to look forward, to motivate you, because otherwise you don't get up. So you need stress, but those are positive. But you have a negative one. So the negative one, you have to practice, and you can start anytime, how to make it positive. So I was terminated in my position several times, even from Temple University. I was subject for deportation. And so that it is always some other people's decision when you were terminated. Other people are saying that you are not good or whatever reason. And then I want to make sure that you don't focus on that and start to feel sorry for yourself, that you work so hard, licking your wound, but rather immediately focus on what next. What should I do now? Because that will be your decision. And then you define yourself and don't let others to define. So that's you have to practice. Okay, I am not saying that you will be kicked out tomorrow and you say, oh, that's good, maybe it's great future for me, but you have to focus on what next, and then try to do the best out of it. And it is also very important that always what I was asking, that what I should do, not like other people should do. People should accept my paper. People should accept my grants. They must be dumb or something. Whatever I say, it won't help. It has to be always and what I should do. I should write better, because if they say they didn't, if I conclude they are dumb, maybe they didn't understand because I didn't explain well. So always had to end up what you have problems is that what you should do, Okay, if the stress is too much or something, then you should walk away. Or, always you have to, what you should do, not your neighbor, your wife, complain about your children or something like that. So it is always, that's the important things, not to blame others, because it won't help you, and not to expect others to do something. You have to do something. And so this was, and that from outside, it seems that, I had all of these failures. But when I was in the laboratory, even when I was in cardiology, I was terminated there, I could see that the experiment's working. Every time I could see the improvement. So it is, you have to feel that you have progress. There are outside progress measuring that how many grants you had. Okay, I get zero out of one. I get only this business grant. I got that one, but never ever anything else. And the papers and others, what they measure on something, but you have to always realize that what you are doing in the bench or as a physician, that you see that you are doing good, and you have to have this confidence. Now that was a long answer for a short question.
That was a great answer. We have another question. Somebody is saying, you worked in multiple labs for decades. Can you explain the process and fundamentals of research design for clinical trials?
Oh, so I work in academia, and it seemed that I was not successful. Yeah, I mean, 10 years ago, I was just sent away, that I never get any money, get out now. So that I went to the industry, and actually it was also at the industry kind of, I was like a basic scientist, have to improve the RNA and have to do animal experiments and whatnot. And I might say here for in the academia, the measure things seem to me is just more paper, positions and other things. What was the measuring? And I found that in the small company, what is measured was more meaningful, because you have to produce something which is, an independent trial is showing beneficial effect. And if not working, then everybody goes home. And also it was like working in a company was that everybody is working together. It's not like you are competing because, and immediately you didn't started the experiments and already fighting who's the first author and last author and whatnot. There was no paper. We just have to do the job, and has to be successful. And so this was the clinical trial. It was mostly designed that how you inpatient escalation you do, or something. It was as a specialist, mostly MDs were doing that, based on maybe on animal data which we generated to design and define that what should be the primary and secondary goals, and to reach those things. And so those are another expert field. I was not part of the clinical trial protocol too directly. I was always try to make the RNA better. And of course, when I went to the industry first, we spent 2 years to make the same thing we could make at PED, but now that is scalable, because what we were doing just precipitated the RNA. It was okay when you had a couple of micrograms, but when you had to make a big amount, you just cannot precipitate. There are, protocols that you have to follow, different one. Scalable.
I think for a follow-up to that question, in your opinion, do you think it's more preferable to work in biotech industry or in academia for students pursuing a PhD?
I mean, in academia, to get to the good lab, you have to have I learned from my own thing, because I never get money, because I came from an unknown country, unknown university, never had a famous supervisor, so why they should give any money? And then I'm proposing something which is too weird. But if you go to a good laboratory and good place, and you can learn from the fellow postdocs and students, you can learn a lot. And in a good university, I mean, that's what I can see actually why in the United States the universities are so good, because in the universities who are doing the research, the forefront, who is just making the discoveries, they are at the universities. In Europe, like in Germany, they are a research institute, and they are not teaching. Who's teaching is who is at the university, who was teaching what they learned, 10 years ago. What they learned, that's what they were. But the best thing is, that if somebody is at the forefront. But if somebody can take the risk to go to in a biotech or something what he discovers and he believes. I mean, that's a bigger risk, of course, because, they can lose support. And you can see that in these days, like just in 1 month, I don't know how many small company was closed down in Massachusetts. So it's no guarantee, because these are just testing out things. And we mostly hear about those which are successful, but yeah. So that it depends also on the person. I never had more than 2 years or 1 year to look forward that I have guaranteed position. Yeah? I never had. So that some people cannot live with that. They need stability. But going out to your comfort zone, taking risk is a good thing.
Dr. Kariko, how did you manage being both a woman in science and a mother?
Yes.
And raising an two-time Olympian.
Yeah, so that I was presenting somewhere, and a student asked that, how could I be, mother, if I would be a man and would have 10 children? If anybody would ask me that, how did I manage? Everybody would assume that, oh, probably, that man has a wife and the wife is taking care of the children. They never ask for a male presenter, but and that's also want to emphasize that. Okay, so my daughter is a 2 times Olympic champion, and so she graduated Ivy League. She got the MBA, and so she was motivated. And I did not assist her. I was 6 o'clock and was in the laboratory. She had to get up by herself, dress up. She learned these things, that responsibility, and that's what I would try to suggest to other parents and scientists, women, that you don't have to over assist your child, and you don't have to give up your dreams, because at the end of it you would say that your child never turn out what you want. They are doing something else always. But so it is important that not over assist. So just to learn, they will learn. I have to say that I pick her up, and she has in her pants and was big holes in her knees. At that time, it was, my daughter is 40 years old, so that when she was in an elementary school, was pants with big holes was not fashionable. Maybe scientist women figured this out. Oh, holes is good. So it is, everything is fine, and you have to also accept that you are not perfect. You don't try to be the perfect sister, mother, daughter. Every role we had, and yeah. So this, yeah. I might say here, that when I was working at University of Pennsylvania, they always introduced me that, oh, meet Kati. Did you know her daughter is 2 times Olympic champion? Nobody said that. Did you know she works with mRNA? That was not the news for anybody. But now I was everywhere. We went to she was 5 times world champion and whatnot. We went everywhere. They said, okay, she's Susan's mom. I was the mom. And now that Susan is coming, my daughter, to the award ceremonies, they said, oh, she's Kati's daughter. No, it is.
Actually, I heard on ESPN your daughter say that now she gets to walk around and say, my mom co-discovered the COVID vaccine.
We have one.
There's a saying, game gives credit to game, which means if you're accomplished, you recognize other people. So I think that says a lot about your daughter, that my mom co-discovered the COVID vaccine.
Of course, that everything, I mentioned several references here, but lot of scientists contributed to all of this science that we are here where we are, and every time when I get an award, I thank all of them. Many people are not live anymore. They published things a long time ago and they pass away, and without them, we would not be where we are. And when I was doing all of this research, I did not expect it in my lifetime that, I would be recognized for anything. That's the other thing, to be humbled. And so that I never crave recognition that people will say my name or they will know who I am. I was happy. I knew that what I was doing. And so that also can keep you calm and not too stressed over that. People are saying, oh, they discovered. Okay, that doesn't matter. The important things they are doing. And I didn't care that whether Moderna said that they discovered everything. Who cares? They are doing, and this will be good.
Well, Isaac Newton's famous quote is that in science, everyone stands on the shoulder of giants.
Yes.
I think to build off that, we have one student who says, my immunology professor contributes much of the credit for mRNA vaccines to you, and it was such an honor to hear you speak about your research. And then why do you believe many other researchers in your field had skepticisms and doubts about mRNA therapy? And what helped you keep a strong conviction in your research despite the thoughts of others?
As I mentioned, in the 90s started, everybody was working with plasmids. And if you ever work with plasmid, you know that you have to isolate from bacteria, and the bacterial RNA is there and the weight is the same. So you add RNAs to this mix, and then you already contaminate the whole laboratory because the RNAs is resistant. If you boil the RNAs, which degrade the RNA, you boil and then it reform, and again it is functional. So they used to say that you have to demolish the building to get rid of the RNA. And so when I told somebody that I am working with RNA, they said, oh my God, everybody felt sorry for me because they could experience that if they try to isolate or make RNA, it is very quickly degraded. And so that was their people's experience. It was labile. It is not really that labile, that and also actually I never work, I don't show here pictures, but I never wear gloves because people wearing gloves and they forget that, oh, I have gloves, and then they put their heads up and then they open the tube and they touch, and the RNAs is everywhere. And so, but if I know that I don't have gloves, I will be always very careful. So that was one thing that in the 90s, from 1990 to 2003 was the Genome project. Everybody was working with DNA, and the idea was that, oh, there is this gene, it has a mutation, and we will deliver with a virus, lentiviral system or something, or at the beginning, we will deliver the correct genes, and then it will be permanent. mRNA is just transient, but now we know that, like Cas9, you don't want that to be around for a long time, and you can make even gene therapy permanent changes using mRNA, like the Intellia did, delivering mRNA, guide RNA, mRNA coding for the Cas9, and did the changes. So just thinking differently, but that was the reason, because it was difficult to work with RNA. It degraded, but most of them because the technology. So everywhere I was putting out sign RNA is free area.
We do have a question that has come in. It's actually a long question. Can you help elaborate on why the SARS COVID-2 spike protein was selected to be the sole target of the mRNA vaccines, rather than incorporating multi-epitopes to help the human immune system recognize emerging variants of the virus, knowing that it has a high mutation frequency?
So mutation was at the beginning when it was created, was not an issue. Yeah. I mean, there was just one sequence information there. And we already know that when new mutant emerged, that those were also incorporated to the vaccines. So that why everybody immediately was focusing on the spike protein, because this is on the surface of the virus, and this is how you want to make neutralizing antibody response for it, and that's how you can manage it. If you are incorporating proteins which will be after infection translated from the RNA, that it was coming to the later stage. And because the mRNA translated inside the cell, so you get a very good cellular immunity, so T-cell immunity, which recognize not only the free virus, but recognize the infected cells as well. Of course, I didn't mention, but many of the vaccine, which is under development, like I mentioned, herpes simplex, those are multivalent. So they are containing 3 or 4 or even more mRNA coding for these different 4 or 5 different proteins. And even now that Moderna's clinical trial is one which has not only the influenza, but the SARS COVID-19 specific vaccine. And even it has a one which had the RSV. So that one injection, that's what how experts see in the future, is not even, you know, multivalent vaccines, but, you know, targeting many different viruses and one injection. But really the you know, for influenza also, that other mRNA coding for not only different variants, HA, hemagglutinin, but other proteins are included. But at the beginning, that was, you know, was quickly developed for just one, the Wuhan sequence for the spike protein. But now that additional proteins are encoding RNA included.
We have a couple questions from students that are biomedical engineering students and hoping to get involved in mRNA research. Do you have any words of wisdom for students that are looking to get their foot in the door in this sort of research?
Oh, so there are so many companies are forming, and the benefit was, you know, as I mentioned, even before the pandemic, there were already mRNA was in clinical trial, advanced trial, and was used for vaccines and other things. It is just accelerated in a way that realizing that the benefit, and then more money was get to the field. And the large companies are also having RNA units, and also many small companies are formed for circular RNA and many different kinds, so that it will be easy to find a place to work on the mRNA field, and it seems like I am very positive that it will be exciting, because it is so easy, so quick. You know that when I started in 1990 doing an experiment, and I just have to describe to you that so I had a cell line which had the protein I was interested, and I just had to isolate the RNA, reverse transcribe. There was so-called TA cloning, as you know, with PCRing, TA cloning, and then I could make an RNA, because all of these things, like I mentioned, 1984 was the SP6 RNA polymerase. On the same year, you could already purchase. That was also in the 80s. That's something you immediately purchased. Or Chomsky made this caltropic agent to make easy to make isolate RNA. I mean, if I would met that guy, I would hug him, because prior to that, it was so much struggle to isolate RNA. And then, so you had this cell line, and then 2 days later, you had an RNA, you put on the cell, and half an hour later, an other cell or something, you know, can make that same protein. And you could analyze that protein in another cell which never made that protein. I mean, it was so powerful. And you didn't need expensive equipment. You vortex and the gel apparat and some very simple things. You sit on the bench, and then you could do all of these things.
We do have an interesting question. It's simply, can we heal cancer more higher by percentage, at a high percentage by vaccine?
Yeah, so that the only field where the messenger RNA was actually used, the first company was Merix, which was spun out from Duke University, 1998 or something. So that messenger RNA was on that field. So cancer vaccine was how Biotech, how CureVac was formed, that everybody wanted cure and make a vaccine for cancer. And the problem was that you identify mutations, especially, melanoma was the most favorite because most mutation accumulated there. But what happened, and then the mutation caused the amino acid chain, and then it would, would show up like a foreign protein. But it seemed that many of these mutations were just not the driver mutations, so that you generated the vaccine against, and you could see that the patient responded, the patient had cellular immune response, but still the tumor was growing because, the stem cells could not eliminate it and so on. And but every time, this trial went on, you always learn from it. The other thing is that, for cancer, rarely, I mentioned these cloning molecules, but rarely you have a protein, a few full protein on the surface of the cell, so that it is easy with antibody to recognize. Most of the cancer is, the cell, it seems normal, and only something is ongoing. Sometimes, you get one extra chromosome, which pushing the cell to divide, and there is nothing abnormal about it. But the other thing was that this epitope, because that's what we recognize, and that's how, the BioNTech and Moderna has individualized cancer vaccine, where they're taking the patient tumor, sequencing, identifying which are the mutation that results amino acid change. Those epitopes are encoded in the RNA with linkers, and several of these strands are given to the patient, just for that patient, and they generate response to these epitopes. But I have to tell you also that 1/3 of the epitopes, which is in our body, is generated, is actually by splicing, peptide splicing. So it is very difficult to see that whether from one protein, take one part from here and one part from the N-terminal, and then maybe it linked to these two little peptide this way together, so that more knowledge is needed to identify what should be the epitope. So it is not just you have a protein and mutation, and that's what the cancer cell is presenting, but it is also can be mixed up, and then it's very hard to predict, even if it's in the same protein. But because sometimes, they found that, what is this epitope? We don't have this protein in the body, and just turn out that, it's spliced, peptide splicing.
We have one question. Somebody's saying, I have to imagine that there was an immense pressure on you and your colleagues during the peak of the pandemic. How did you deal with that? And did you ever feel discouraged or rushed?
I came on March 13, 2020, I came home because I was living in the United States, but at age 58, I went to Germany. People thought that I am going to the wrong direction to the industry, but anyway, I came to the United States on that night when the country was closed. So I was here in the United States. I couldn't fly. There were no airplane, flying. And my colleagues who were there, of course, we had a computer, and most of the things which my team, I had finally at Biotech, I had a team, were, to figure out what should be the formulation, what should be the proper sequence on the untranslated region, which makes that least RNA, we get the most protein, and so on. And those were done in 2013, 14, 15, 16. So it was, and it was already, all of this collaborative agreement between Pfizer and BioNTech was already signed in 2018 for the influenza vaccine. So we already worked together. So everything seemed like we can start the human trial for influenza, mRNA-based influenza vaccine. And so we already knew that what should be the formulation, what should be the modification, and all other things. So it was just a switch over, and it was the role was to develop assays to measure when the antibody is coming, measuring things, so that you have to develop assay, you have to develop pseudoviruses to see, the reaction and develop assays. So that was mostly what had to be done at that point. But we already knew what it is. And of course, what is the biggest challenge was, which is obvious, is that scaling up. Because you had the one grams of lipid, and tomorrow you need 1 tons of lipid. And this is, what we usually say, that when you have guests and you are cooking for 2 persons, you can steer it and and then tomorrow is 200 people is coming, is not enough to get a bigger pot, a bigger stove, and a bigger spoon. Because you can steer, but you never could make what is in the bottom is burning and the top is still cold, because you have to introduce a new technology. Maybe you have to put everything in a tubing and burn under, like for the food I mentioned. But this is, you have to come up with a new solution, that how I can, do it in a large scale, which nobody has ever done. And these are not in my expertise. There were for everything. There are so many experts. I mean, for thinking about Pfizer. Okay, this is a polymerase. In 1 vial, we have to have multi-dose. And Moderna, which had less experience, is not a big company, didn't know that, and they ran out of glass vials because it was individual. Now it was multi-dose vials. It figuring out that we need aeroplane, we need to CO2. We don't have enough CO2. The dry ice, and then we have to make quickly a company or a not company, but making a unit to produce that. And then they realize, oh, there is a limit how much dry ice can be by an aeroplane. Oh, somebody has to look it up and then change it. And then, all of these people are just know what to do. And running the clinical trial, it is also that, the big companies knows how to do it very quickly, smoothly, because they have done so many times, and all of these experts are there. And this is their responsibility, somehow. And many people emailing me that they want to get to the Biotech cancer trial and different things. They don't know that the company is independently, the trial is running. We don't say that who should go there, because that's the independence. That's the, that everything will be objective. The sites will sign who can enter to a trial, what is the criteria, everything. It's not the company will tell that take these people and not to take, but most of the people, and that's the problem, and that's why we are here to educate the people, the public. That was missing, and then I mentioned in the beginning that we said that we have a product, but we never worked the society to accept it, because we did not explain to the average person that how this works, what is this RNA, and then they listen to those people who had time to go to the studio and didn't have to work out how to make, things work, and they were, the real experts were working in the laboratories. But those who had time, they went to the studio and they said so many stupid things because they could say it so calmly and so convincingly, and then scared people.
Another long question we have is, knowing that you have been steadily working to develop mRNA technology for roughly 30 years, do you feel confident that this technology was ready to be widely applied to develop and improve established vaccines, emerging vaccines, gene therapies, and also targeted drug therapeutics prior to the pandemic, that is?
Yeah, as I mentioned already, prior to the pandemic, it was already tested out, used. It was, was difficult to get money, but if you would attend 2013, we have mRNA Health Conference where just the in vitro transcribed RNA is used. There are companies were already in China, the Stamirna, there were Akurna in Japan, there is South Korea, they are all over the world. Germany had several companies you just didn't know about because you were not on that field, that all of these different companies are going in different direction, different therapies, focusing it is interesting because when I talk about, the mRNA therapy, prior to that, it was always the problem was that, oh, that's so labile, never will, immediately degrade. And all of a sudden, when people started to be afraid of the vaccine, oh my God, it will not degrade, the RNA will be there forever, the protein. It was all of a sudden the opposite direction came, that it because the people didn't know. And they did, that when also this ask about these effects and like, that it will incorporate to the genomes and other things that the people didn't know that the messenger RNA is never going back to the nuclei. You need a special virus protein. And even if it would go back, that it has to be reverse transcribed. Okay, if there is a reverse transcript there, the enzyme still need primer. You cannot, DNA cannot be synthesized without a primer, and primer need complementarity. So there are so many if there that but the people will not explain that, and they ask me that explain to them that it's not happening. I mean, if you believe that it can happen, for example, that's one, then you don't know enough. And then the end of it, I can explain why it and whatever is required to incorporate to the genome. But, whether I will be sound convincing, it will be just by belief and not by knowledge. Because the gap between average person knowledge, and where we are is so huge. And somehow we have to close that gap. And somehow we have to talk, and that's what, during now getting awards, I try to explain some simple term, find analogy for something to explain how something happens. But what was the question? It was about that. Yeah, but I believe that it will be therapeutically and as a vaccine. I mean, with colleagues here, Drew Weissman, Norbert Padi, in 2007, 16, 17, we already with Zika virus, identical mRNA modification, cap, other elements, formulation. It works so well in monkeys that we could see that nothing compared that how powerful the mRNA vaccine. And what was surprising, you guys, is that very small amount was sufficient. So in mice, we tested out 30 microgram, and 50 or the same amount in monkeys was also effective. Prior to that, when people tried to use DNA for vaccination, unproportionally, they had to give more, like milligrams, when the body was like in larger animals or in humans. So that was unproportionally had to increase the dose. And here again, like SARS COVID-19, the same 30 microgram was good in mice, 30 microgram was good in human.
mRNA therapy is just an absolutely incredible accomplishment and a breakthrough in science. And I was just wondering, what do you think in terms of challenges, what are some of the challenges that scientists such as yourself face when introducing a new groundbreaking therapy? And how do you deal with those challenges?
Yeah, so that it is 50 years the molecular therapy started, 50 years, which was first time, restriction enzyme was cut, the gene and put in somewhere else. And of course, the scientists were there. This meeting was called that we are scientists are concerned about something, and we have to make sure that everything is safe and good. And what was about that? It was the safety. What was the question?
Just the challenges that are faced by introducing a new It's not new, but new to the public.
Yes, because, we can say that, we did not invented RNA, messenger RNA. It is in our body, but still we are delivering on a way that not naturally is delivered. Or now we also learn that some exosome is delivering mRNA in our bodies. But this is one thing not to make any harm, for all scientists is there to make sure that whatever we are doing is safe and reliable, well understood, and so that's one thing. The challenge is for the future wise, that how this whole thing will. So if you can target specific cells, tissues, or in organs, specific cells which you need to introduce protein or something which is needed, and you are deciding to do with mRNA. So that's the future is that to make targeted delivery of the mRNA, and that's what many is working, and that would be the future, so that you could selectively deliver to we can deliver to the spleen, for example, what we are doing, or the hepatocytes in the liver, or we can put delivery method, specifically I mentioned for T cells in vivo can be, now that bone marrow also can be delivered, and so that you can do beneficial changes in some people who suffering different bone marrow related diseases, hematopoietic cells can be changed. And so that will be less side effect because you are targeting specifically into those cells. So that's the future. You will get more in it. But when you discover, like the CRISPR, also that people are, concerned about whether there will be some problem with it, and then the scientists who are doing, they are the best to judge that what kind of things can go wrong, and then they can make sure that it won't happen. And so we have to trust that the scientists, Paul Berg, who just passed away recently and got Nobel Prize, and he did a Olisome conference, was, when he was aware that maybe people are worried about, everybody worried, and make sure that, maybe some altered bacteria will not get in our system or something, and to make sure that we have all of the safety there. So I believe that the scientists are good at checking on those things.
Did you naturally have an aptitude for biology, or did you have to put in a lot of hours of studying? And if the latter, can you share some tips that you personally utilize to maintain your grade and retain the material?
I always like to read, and elementary school, I was third best in the country, already competed in 7th and 8th grade already. And I have to say that I spent hours, even I am giving lecture, but even preparing myself. I want to make sure on this presentation to make sure that I check out everything, that what's happened. Maybe I missed out something. I even talk to my colleague to make sure that every virus will be listed, which is already entered clinical trial, and won't tell you some information which is not true. And so to be on the top of things is difficult. I showed you in the 1990s, every year one paper. That was easy to follow. Now that when you have every hour you have 10 papers, then it is, this 1 hour, I just missed that 10 papers. And so it is, I have to say, coming to the United States, the big advantage I realized then is that I could listen great talks. And I am talking in the 80s. I remember 1 hour we just drove to to listen Bob Gallo, who was talking about HIV and the patient, and there were no internet, nothing, that you had to go and listen and drove 1 hour and listen there. What's going on? What is this virus? Why is going here and there? And then it was here at the university, every week there were like, every day I could sit on presentation, but I had to limit myself at least not more than 2 in a week. And I prepared, I was reading what these people were doing, and okay, I will go that and that. And I selected scientists that were not what I was doing. I was just thought that, oh, that's very interesting. I never heard about here to learn. So that now, right now, you can listen on the, YouTube on many, now that maybe the difficulties is who to listen. But there was, that was the learning process. I was reading and thinking about that. This is very interesting. I never heard about. So that was mostly the learning. And then I looked it up, that iron sulfur cluster protein, what the hell is this? Oh, this is how we put our iron in our enzyme. I didn't know that. And then I was just keep learning. And that's how it is so exciting. So I can sort of comment to the students that please go to lectures and listen to, even if it is not familiar the topic but for you, but if in advance you can just read about what is all about, that it is, you can learn so much more. It is like going to a country and not speaking the language. When you come back and start to learn, you don't remember what they said because you didn't understand anything. But if you start to learn in advance, you can learn so much.
You mentioned a lot of different clinical trials going on, and also the targeted approach to having mRNA target specific cells. I was just wondering, what are you most excited? Which future developments for mRNA therapy really are you excited about and look forward to seeing?
I mean, mostly in protein therapies, which I am more interested, not the vaccines, but I mean, I resigned my position in October in Biotech because I want to focus on something which actually is not even messenger RNA, which sounds like, because even prior to working with messenger RNA, I was interested in other fields. And then I thought that I always wanted to do certain other things. But on the messenger RNA field, of course, these different therapies, which I am, want to see that how it turn out, and I am rooting for many clinical teams.
Are there any best graduate careers that you would recommend someone to pursue if they wanted to eventually end up in your specific field?
I mean, I am by training biochemist, but you have to have a field. I find, the immunology is the most appealing. It is, you have to learn immunology, and then you can go to many different fields. I find that. So immunology is, again, when I met Weissman, I told him that I was not worried about overexpressing a protein for the stroke patient, because those proteins are already in their bodies and they are not foreign. I didn't know that everything changed from when I learned immunology, and the immunology is, is not seeing foreignness. If you inject the spike protein, nothing happened. You have to have a adjuvant. You have to have something to tell the immune system, hey, pay attention on this one because that's dangerous, And so but that was the same when I went to the lipid team, and I still learned that, there is protein, the sandwich model. And then I go there in, a couple of years after I finished high school and in a lipid lab. And then they said, oh, no, that this is a mosaic model. The proteins are actually go through the lipid. And, you just constantly learn that what you learned is, outdated. And constantly you have to learn and be on the top of things. And so immunology, I found the most exciting and appealing field, which I was not too good at the beginning, but I keep learning and reading about. And that's the other thing, is difficulties to remember all of these papers. When you read 10 papers, you just kind of remember 100 and 1,000. And you can put in a folder. But what I did a very nice Excel sheet, and then I collected every mRNA, that what was coding, how it was tested, what was injected, or it is human trial and everything. And every time I just read a paper, that just one line in this, Excel sheet, and then I could looking for something. I didn't have to go to different folders, try to find that paper. It was just like, I look at my sheet and then check it out. Now I remember. And also I put there in an abstract of the paper and also the EndNote link, so that I can, if I write a review, then I can just copy and paste. So did little systems, like a little bookkeeping.
I know we're getting close to the end, and I don't want to keep you too long, but we do have one question. Somebody is asking, how did you and your colleagues obtain grants and funds for research? And could you describe the process?
Yes, so I convinced Elliot Barnaden when I was in cardiology that RNA is good, and he could apply for grants for other things, and then how he supported my salary then. And Drew we did a lot of Drew Weissman, for vaccine, try to send in, but it was many times was not successful. So that from the NIH. And there are also, the university has a smaller, that most of the university has internally has small grant. You have some idea, and then you can get like $20, $30, $50,000 just to make the prior experiment enough data generated to apply for a bigger grant. Even if you want a business grant, and as a university, we first apply for a Pennsylvania little grant, so that we can set up a business plan to write and get enough to apply for a small business grant, which was the STTR grant or SBR grant, and different grants from the government. You can have money. You can have also companies' money. They have smaller, if you have specifically some disease-related proposal. But so usually universities have internally have some grants that twice a year you can apply and get some little startup money for your ideas. I am not the right person to ask how to get things, because I was not very successful. But if you can convince your colleague who is successful to get money, and then he supports you, then that's also one way. That's how I got my money or my salary. But you don't have to think about, I started at university in 89, 90. Then my salary was 40,000 as a faculty. And then 20 years later, I get 60,000. So in 2010, I get 60,000, and the technician get more, but it did not matter, for me. So that because, that was my hobby. I didn't have to spend my money on some hobbies, because I enjoyed. That's what I wanted to be, there in the lab and thinking, and many weekends that I realized that if I go on Saturday, then by Monday I learned something. So I always figured out that, just quickly, like 2 hours, I will go there and I can accelerate things. Many times my colleague mentioned that I told that, I wish I would be 1 week older or 1 month older, because by that time I would know the outcome. I wanted to be always older.
Dr. Kariko, you came to the US from Hungary as an immigrant.
Mm-hmm.
How would you describe that feeling of returning back to Hungary as a recognized national hero?
So people, not just that one, they many times ask, how about those people who, showed you the door? But I never had, that's also good advice, to revenge or fear something like that. No, as actually, I was receiving an award in Canada, the International Gardener Award. Thanks to all of those people who tried to make my life miserable. Because without them, I would not be here. I would not be fighting stronger, because those who are fighting against you, making you work harder, and you improve constantly. If your conclusion is not that, first of all, comparing yourself to others and realizing that I work so much more, and I'm not advancing, and I'm seeing the others working less, and don't ever compare yourself to others, because you already took away your attention. What you can change, you cannot change that somebody else is the favorite of the PI or something, or you feel that they are not working. Maybe they are reading much more. You don't know, just don't even bother, because you will get disappointed. And if you focus on what you can change, your experiment, reading more, listening more lectures, and learning constantly, then you can improve yourself, and don't get disappointed. And I'm going back to Hungary. I am very grateful to all of those people. I mentioned there Eva and Ernő and the lipid team. They were the first time showed me that how to make liposome, and now inviting them to, when I get an award, I inviting them to come over and celebrate together, and I share, because without them, I would not thought about delivering nucleic acid, or I visited Jenny Thomas just last year, who was making the cap analog once in RNA team, and I am very grateful to him. Even he terminated my position because we ran out of money. I don't blame them. It is just, I am grateful that I could learn with RNA, and so much I could learn from them. So that's how I feel. And of course, all of the, fame and, so I am telling you, if you want to be in the spotlight, then go to be an actress, because in science it rarely happens. And I was couldn't find my things, what to say, at the beginning. And then I thought that maybe I have to inspire the next generation. Then I also felt that we have to explain the science to others, that so that they can understand better, and then they can decide. Because many people who were skeptical about the vaccine and others, they had genuine questions, and they, some people, of course, said already what they thought, and then they didn't need it because you cannot convince them. But many people was just wanted to know and didn't know. Yeah.
Thank you so much, Dr. Kariko. This was an amazing session. We've had lots of people thanking you and saying how insightful this was. And I think just to close, we'd like to know if you could give your one piece of advice to your 20-year-old self, what would it be?
Yes. So whatever I gave already myself, first I have to do something which I like, and I like to do experiments. I like I also to believe, coming from a small house, there we had no running water and other things there. And then coming to this University of Pennsylvania, everybody's perfect English, and everybody's publishing every week some kind of nature science, you can feel intimidated. And, just have to believe that you can think about something that nobody is thinking. That's what, you have to believe in yourself, because if you believe that you fail, then you will fail, because then you, that's also that, but these are the things actually I learned from this Shaya, that I have to always focus on what I can do. And so that's what I followed, that so that not blaming others. So finding that what I can do, and not letting others to define me, but when it was difficulties or when they send away, that I will have to make a decision what to do next. And going from university going to Biotech when had no website, nobody even know about. I just accidentally learned from another guy who was also kicked out from another company, told me about this Biotech. I said, oh, okay, I will go and visit them. So it was, yeah, so not always some fancy places. Maybe you will go somewhere, maybe less fancy.
And Biotech was also founded by immigrants too.
Yes.
And finally, one of our viewers would like to ask you what your perspective is on the possible mechanism for myocarditis, which may or may not be a side effect of the mRNA vaccine.
Mm-hmm. I mean, that was the only side effect which was specifically demonstrated when they looked at almost 1 million person in 1 year in Israel, that what kind of disease they got, those who get vaccine and those people who never get vaccine. 1 year, how many deaths for stroke and other things, and it when they compare, that was the only one. This I have to say that those who are saying that for a month they can see they detected spike protein circulation and other things already, it was already the technology, how they determine that was already all in question. So I don't know the reason. I am not a physician, so to know that what mechanism could be responsible. But it was real that and don't also understand why those who are young male is and not as much as the elderly or the young girls would get this. So I don't know the physiology, and I am not a physician would know. I read about different hypotheses what it could be, but the proof. What is really the reason? I don't know, and I just again the same thing for the origin of the virus. I don't know. Could be here, there. I don't know. I focus on what I can change, what I learn, and reading about what studies where data are there, and that's that is not data are there, but I don't have the knowledge. Yeah, sorry.
Well, thank you very much for making time again on Saturday morning or Saturday afternoon at your time.
Yes.
To join us, and we're really honored to have you. We know that you're really busy and you've been traveling quite a bit and receiving awards that I never knew existed. I think I went on this Wikipedia and all the awards that you've won, and it's been very, it's been well deserved and well recognized. And again, thank you so much for making time for us, and safe travels.
Yeah, I might say something that for the girls, if they are there, or the boys who are listening, or the males, that so right now, just like always, my husband happened. Right now, Saturday, he asked me that when he would have to put the things in the oven and cook, he start to cook for our dinner because I will be busy here. And that was the same. So that what I am saying, that when you can have, the family and all of this other, you have to have the right partner who helps you, recognize that you have dreams, and not that serving you, but, and then I could ask the guys be such a partner for their wives and help them out and start the cooking when here the students want to listen to me. And I did not realize it will be Easter Saturday, but now we are here. So thank you.
Yes, and yeah, and I would say for our organization, I'm like the only one of two guys out of, and it's everybody is women. So you know, I personally, I always say I personally like working with women better because they just get things done,
Yeah.
and there's not a lot of ego. But like our organization is it's majority women, and me and another guy are like the only two men in the group, and I think we have 11 other women. So we're definitely, and I think it's the importance of it recognizing that there are challenges and differences, and I think that everybody has a talent and everybody has an expertise. Like I'm the least talented person when it comes to like arts and graphics and that stuff, but everybody has, like, for example, Madeline makes most of our flyers. We have another person, Lucia, that does them. And so, and if you ask me to make a flyer, it would be very bland. It would just be black and white. But I think that working in teams, I think it's really important. And I think we all learn from each other and support each other, which I think is really an important thing. And again, thank you for making time, and thank your husband for making time. And again, it was a really great honor to meet you, and safe travels, and across the world, and hopefully and then again, thank you for all your work in the vaccine. Certainly as a frontline health worker, I could tell you that at the beginning of the pandemic, it was very scary for us. We didn't have gowns, and we didn't know what this thing was. And 3 years later, we are vaccinated, and we still are precautious, but we've gone back. So I think thank you for your team and all the work that you have done, and being consistent and being an inspiration to us all.
Thank you.
Thank you very much.
Thank you
Thank
so
you.
much, Dr. Kariko. This was incredible. Thank you.
Thank you. Take care. Bye.